to record in 3D structures and active liquid interfaces. And today he switched to a more biologically oriented project in the lab of Joe Howard at Yale. So please, Ray, floor is yours.
All right, thank you. Does that work? Can everyone see it? Does it look right?
Looks great.
Okay, great. Okay, let me get this little box out of the way. Okay, so
no box.
Yep, all set. Okay, so hi everyone. I'm Ray, and appreciate you all being here today. It's nice to see so many familiar folks. And today I wanted to talk to you about my project with the goal of studying self-organization of the simple Cnidarian nervous system. So, as means of an outline today, I wanted to first start by talking about animals, Cnidarians generically, and then why I think they're great models and why I think they're great models for studying physics of soft, living, and active matter. I wanted to talk about their role, I think, in studying self-organization in neuroscience, and then round out by talking about some of the experimental work that we're developing. So this last part is going to be pretty heavy on engineering and methods. So if you're not so keen on that, sorry, right off the top. So the goals of this talk really are to first share, I think, this very unique model system that is attracting some attention, but I think is sort of maybe a little underappreciated, and discuss some of the methods that I hope are broadly relevant to folks here. So right from the beginning, talking about Cnidarians, and I wanted to start with the question of what would be an ideal model animal to study self-assembly or the physics of self-assembly. And there's a lot of possible answers, but I think I made a little list here, which is first, simplicity. If you're studying physics of some living system, life is already complex enough, and you want something that is as simple as possible so that we can use these physics models to describe them. You want something that has very clear self-assembly so that it starts in a very clean initial condition and then assembles into some known structure. You'd like to have a model with very clean and clear experimental controls, which for a biological system means something that has nice transgenic lines, and you can do knock-ins and knockouts and CRISPR and all these fancy techniques. And as a bonus, if it's something that's generalizable to other models, that's just even better. And so what I'd like to convince you of today is that Cnidarian fit all of these criteria extremely well. I don't know if I said right from the beginning, but please interrupt and jump in whenever. I don't know if that's how these normally go, but certainly ask any questions at any point. So what are Cnidaria? So if we start here from the phylogenetic tree of all animals, all the way at the base, we have porifera, which are sponges and things like that. Next level up, you have placozoa, which is a really, really interesting animal. It's a single layer of cells that sort of crawls along the bottom of the ocean. If you haven't seen them before, take a look at some YouTube videos. They're really neat. And then next level up is our animals of interest, Cnidaria. These are animals that generally have radial symmetry, so they include jellyfish, corals, and anemones. And then all the way at the top is everyone's favorite, Bilateria, which have left-right symmetry and include almost every other animal. So extremely successful group of animals. And then, of course, because things are always more complex, there's these really weird ctenophora that are probably down here and are extremely complicated, but probably even more primitive than porifera. So lots of fun debate going on about where these are. So zooming in on Cnidaria, there are cultured as laboratory animals well over 100 different Cnidarian species, but there are 4 that really dominate the market. So these are Hydra, which are freshwater polyps. They're about 1 millimeter long, but they change their body size based on how well fed they are. Hydroctinia, which are colonies of saltwater polyps that spread over shells and clone themselves and spread over whatever surface you put them on. Clidia, which serve as sort of a model jellyfish animal, and Nematostella, which are these sort of frighteningly long anemones. They're something like a couple inches, and they're sort of creepy looking. And they live in brackish conditions. So we'll primarily look at Hydra today, because that's what I've done most of my work on, but there'll be a little bit about some of these other animals. Okay, so Hydra and all Cnidaria have a pretty simple body plan in that they're made of two layers of cells, an endoderm and an ectoderm. They really have no respiratory system or circulatory system, no organs, and any other cell types other than endodermal and ectodermal cells are just sort of stuck in these two layers. So I think they fit the bill of being pretty simple animals. There's great experimental controls that have been built. So for instance, you can do simple fluorescence labeling. In this case, they're labeling hydroctinia with GFP in their neurons, and so you can track what their neurons are doing. And then all the way up to much more complex stuff that's still being developed, where folks use CRISPR to knock in like eyelid and these sort of things, so they can do optogenetic induction and express certain proteins and certain cell lines and look at how this affects morphogens downstream. Point being that there's all these very complex things that have been developed in Cnidarians. So we have great experimental control, even though they're quite primitive animals and aren't studied quite as well as so maybe some of the more famous ones. For Hydra, what you can do is we can express in specific cell lines. So for instance, we could express proteins in the endoderm and ectoderm. So here they're expressing a red fluorescent protein and green fluorescent protein in the endoderm and ectoderm. And similarly, you can express things in the neuron specifically, which we'll be very interested in. Okay, so as far as self-assembly goes, Cnidarians are spectacular. So they're capable of regenerating. So for instance, you can take, in general, Cnidarians and cut them into pieces, and each of those pieces will grow back into a whole animal. And this is true across Cnidarians. But Hydra in particular are really great at this, where they can take any section of the body, cut a little hunk, and this will hollow out into a sphere, and over time will regenerate back into a whole animal. So that looks something like this, right? So this thing is closed up, it starts to eject dead cells, and what you'll see is pretty quickly it'll establish a body axis and start growing tentacles and elongating. And so now this thing has regenerated back into a whole animal. And you can go even better is you can target individual cell lines. So for instance, this is an experiment we did where we're expressing fluorescent protein and an activatable toxin in the neurons. And when you activate this toxin, sure enough, all your neurons disappear. And then over the course of about 5 days, they recover. So you can both, chop up this animal and see it regenerate, or you can watch the regeneration of individual cell lines in an intact animal. Beyond this, Hydra are really, really famous for their reaggregation abilities. So what you can do is take Hydra and disassociate it into individual cells. And so this is the original paper to do this way back in 1972. They took Hydra, broke it into individual cells, passed it through a screen so that they were sure it was really individual cells. And what they find is that over time, a few days, it reassembles back into a whole animal. And so if you're interested in, self-organization and self-assembly, this is a fantastic model. More recently, folks have, looked at individual cell lines and how they work during this reaggregation process. And just to convince you that, Hydra aren't really all that special, you can take other animals. So for instance, this is an experiment we did where if you take hydroctinia, what we found is that they are also capable of this reaggregation. So it seems that this may be quite a bit more common across Cnidaria. And so there is sort of benefits to having such a simple body plan. And then as a last point, I wanted to argue that Cnidarian may have more broad relevance. So back to this sort of phylogenetic tree, it turns out that our common ancestor with Cnidaria had neurons, synapse, and muscle. And so anything that you find that is sort of interesting from a self-assembly perspective, it may, have some sort of interest in studying in other animals. Whereas if you look at simpler animals, you may miss these sort of neurons and synapse and muscle. So I'm not the first to point out that Cnidarians are sort of a rich system to study the physics of life. There was one fairly famous paper that looked at topological defects in Hydra.
Great. Can I ask you
Yeah,
question
yeah.
about the previous slide
Yeah.
where you go on talking about interesting things on defects. Given that, and this is partly ignorance,
Yeah.
in bilateria, do all bilaterians have three germ layers?
Yes.
Okay, and so given that Cnidaria have these sort of essentially two germ layers and with unclear sort of non-direct association of what's the there's something that's ectoderm and there's the endoderm mesendoderm sort of partly has mesoderm and endoderm features. What are and given that they have this sort of rather general, as you point out, regenerative ability, are there generic things one can say about the number of germ layers and the loss of regeneration? Is there
Yeah.
something in that context?
I don't know. So, I mean, there are bilateria that can regenerate, right? I mean, there's a few famous examples. I mean, like zebrafish embryos.
Sure, but they're usually timed, right? You don't do it.
Yeah.
You lose that ability at later stages, and so and this sort of rather extreme plasticity and repetitive regeneration, which is extremely and the viable fragments, no matter how small you start.
Yep.
Is there something about the kinds of body plans that don't allow for complex regeneration over and over again, and what kinds of body plans do?
Yeah, I don't know if it's necessary the number of germ layers as much as it is in Cnidarian you have this presence of pluripotent stem cells that just exist throughout the entire animal's life. So that really helps quite a bit. And then there are some that are capable of pretty interesting things like that we don't necessarily see quite as common in bilateria, like when stressed, Cnidaria differentiated cells can go back into stem cells. So there's all these really wild things that happen that really give you this regenerative abilities.
Okay, thank you.
Yeah, sure. Any more questions so far? Okay, great. So then, so there's a fairly famous paper that looked at topological defects on the surface of a regenerating sphere of Hydra. It looks something like this. So you see here a plus half and a minus half, which have been highlighted here. And these will, over time in this video, will be attracted toward each other, meet, merge, and then annihilate. And so for lots of folks here who are interested in active nematics, I think this is sort of an interesting feature. And what they had found was that these topological defects sort of predict morphology. So +1 defects predict where a foot or a tentacle might be, and -1/2 defects predict where certain parts of the head are going to end up. And more recent work has looked at mechanical confinement and strain and how that biases defect ordering. And so what they had found was that when you squeeze certain part, you produce certain defects, and that then predicts formation of different features. And so there's really interesting and non-trivial physics that's happening here between mechanical confinement and defect ordering and morphology. That's still ongoing. There's lots of folks looking at this. Beyond that, there's folks looking at like morphological changes like this, where they have a sphere that then breaks symmetry and becomes this elongated tube on the way toward regeneration. And they look at this as like sort of a first order like transition. As you change different conditions, you can bias it in one way or the other. Lots of interesting stuff on body dynamics, including like mouth opening and how for Hydra and these sort of polyps to open their mouth. You have to use a model that's like this ball and spring with very nonlinear elasticity, or studying the locomotion, where folks find that tissue stiffness gradients are important for behaviors like somersaulting, where this thing goes head over foot as it escapes some noxious stimuli. So lots and lots of really interesting physics that is being done and still to be done using Cnidarian. So I was going to jump now toward talking a little bit about Cnidarian neurons. Is there anything else, any other questions before we move along? Okay, great. So, I wanted to start with a very broad question, which is, how does the nervous system self-organize? And this is a large question with lots of possible answers, but I think one such answer has to be as synchronous groups, which are called lots of things, but we'll call ensembles. So here is an image of neurons in a mouse, and what you see is that there are these flashing. These neurons are expressing GCamp, and so when you see a flash, that's a neuron firing. And so what you see is that there's these large-scale synchronous firing of lots of neurons together. And there's lots of work that's studied these sort of synchronous groups. And what they find is that, these groups seem to be associated with distinct stimuli, and that you can elicit certain behaviors by stimulating these groups. And so you can, for instance, go in and activate certain neurons and cause a group to fire, and then cause a known behavior. In this case, they were doing kind of mind control, where they're activating a group that causes a mouse to stick its tongue out whenever they activate it. So these experiments and many others have led people to think that maybe these synchronous groups form some sort of basis for neural computation. In any case, they seem to be incredibly important, and they exist also in Hydra. So again, this is Hydra expressing GCamp, and what you see is these synchronous flashes throughout the body. And so because Hydra's nervous system is so simple, it's very sparse and open, unlike ours, where everything's condensed together. The nerve net in Hydra is spread out over its body. There are really only a few hundred neurons and a few cell types. So there's hope that this thing is simple and may even be tractable. You might be able to understand what's going on during the formation and development of this nervous system that you may not have access to in more complicated models like us. They still, even though they're simple, display non-trivial synchronization states. So just from this video, you can see different firing, and different neurons are flashing and then causing another group to flash. So there's lots of stuff going on. So to say just that they're still interesting to study. So the question we're going to look at is how do neurons synchronize, and specifically how do they synchronize in Hydra? And there are lots of possible models that have been put forth for neural synchronization. The simplest one is just to use a simple harmonic oscillator, which, again, the simplest model is using something like Adler and Kuramoto synchronization to drag your two oscillators and cause them to synchronize. And this is used for all sorts of biological systems, including like swimming of Chlamydomonas, and lots of other things. But you may say that that's not good enough, that to describe neurons, you really need something more complicated than a simple harmonic oscillator. Maybe you need something that integrates then fires. So, no problem. There are lots of models that exist like that are used to describe things like synchronization of firefly flashing. You may say, well, that's not enough. You need something like Strogatz or FHN model or Hopfield model, or maybe you need to consider connectivity or polarity of the neurons, or all sorts of other things. The point being that there are lots and lots of synchronization models, but there's not a lot of experimental data where we're watching large groups of neurons synchronize together and testing these models. And so that's really the goal, is to use Hydra to test these sort of models. So as a neuroscience model, Hydra's been developing in the last few years. First is that folks have started this sort of functional calcium imaging. And what they found is that these neurons fire together in distinct synchronous groups. So for instance, this is a group that governs body contraction. So you can see there's lots of neurons here in the foot, but some throughout the body that when they fire together cause the body to contract. Same thing here is another group that governs elongation. And generically speaking, they find that the entire nerve net is is assembled into ensembles, and these distinct ensembles encode specific behaviors. So there's one group that always fires together that causes nodding, or there's another group that causes contraction or elongation. And unlike bilateria, it seems that these primitive nerve nets work in a way such that there's a one subset, one circuit. And what I mean by that is that if you're a contraction burst neuron, you always fire with contraction burst neurons. You all fire together, and you all together produce a single behavior. Whereas for animals like us, you might have multiple neurons controlling different behaviors, or a single behavior controlled by different subsets of neurons. So it's much more complex, and here things are extremely simple.
So they also simple in size, like the cluster size scales differently with the task.
Sorry?
The cluster size, you say that the subset groups always fire at once. Is it always the same size of a cluster as well?
So if you, yeah. So do you mean like between different animals, or I guess I can answer both ways. So the animal itself, so an individual hydra is effectively, lives forever, right? And it scales its body size based on how much you feed it. So if you feed it a lot, it grows pretty big, and its number of neurons increases. If you feed it very little, its body will shrink, and you'll have very few neurons. What you find is that the proportion of neurons, so like the proportion of neurons that are associated with contraction, is about the same whether the body grows and shrinks. Now, you can change the number of neurons by up to an order of magnitude depending on how much you fed it and the temperature it's cultured in and that sort of thing. But the proportion remains the same effectively. Does that answer your question?
Yes, thank you.
Yeah, sure. But then between groups, there's very different number of neurons. So for instance, the one that causes nodding or bending of the body is very few neurons compared to like this contraction, which is very dramatic. Okay, so then back to looking at the aggregates. In Hydra, you can do this spectacular reaggregation process. And if you reaggregate animals that are expressing these fluorescent calcium proteins, GCAMP, what you find is that initially, 8 hours after you first form your aggregate, the neurons fire in an uncoordinated way, meaning that each neuron fires whenever some internal biochemical clock tells it to fire. But at about 72 hours, you start to see groups of neurons starting to emerge and starting to synchronize. And then by about 8 days, you have an animal that in its body plan is still very strange. It has multiple heads and multiple feet. But if you're looking at the structure of the nerve net, it's almost identical to what you see in the adult polyp. And so the question we had started with is, how does this functional architecture of the nervous system emerge? And what we'd like to do is really just watch this resynchronization over time. So starting from an initial aggregate all the way through to the full redevelopment of the nerve net, what happens and what physical models describe this sort of resynchronization. And so from there, I'm going to move to some experimental work that we're developing toward this goal. But again, I'll pause and see if there's any questions. So any questions now?
So what's meant by functional architecture, or is that something you're going to cover?
No. So what I mean is, how do you get these different structures, or specifically, how do you go from a collection of mixed up neurons where you have them initially uncoordinated to these sort of stereotyped groups where you have these contraction ones at the bottom that all fire together, and these elongation ones that are distributed throughout the body. How do you emerge this sort of synchronous group, and how do you emerge with these 4 synchronous groups?
Thanks.
Yeah.
Hi, I have a question.
Yes.
So these groups, are they kind of throughout the body? Are they mixed somehow, intertwined, or not? So either are they physically separated? Yeah.
Yeah. So it's a great question. So so you have the endoderm and the ectoderm layer, right?
Mm-hmm.
Between them, you have a basement membrane that does separate the endoderm from the ectoderm. And what it seems like is that's the only real physical barrier keeping groups of neurons apart. So there are some that are in the endoderm and some that are in the ectoderm. So that's the only way to really divide the cells from each other. Inside the endoderm and the ectoderm, you have lots of neurons of different groups that are overlapping. So what I mean is that like contraction burst is associated with neurons that are in the ectoderm, and so are rhythm potential 1, which is associated with elongation. They don't seem to interact with each other, so they don't fire together. So you may think, well, maybe they're not, wired together, and that's what it seems to be the case. But physically, they can be in contact with each other. So there's some, molecular cue that tells them when they should fire with who. Yeah.
Mm-hmm. Thanks.
But there is a distinct internal and external nerve net that are separated by this basement membrane.
Mm-hmm.
Yep.
Thanks.
Yeah, sure. Any other questions?
Yes, I have one question. Just about the length scales in the
Yeah,
system.
yeah.
So, what are the sizes of the neuron cells, and how far are they apart from each other?
Yeah. So it depends where you are in the body, but just as a rough number, the body of the cells is going to be, let's say 5 microns. The distance apart from each other, let's say, is something of order like 10 microns. Again, if you're very near the base here, things might be closer together, and if you're further out here, things are further apart. So the density of the neurons changes across the length of the animal.
So it's easy to distinctly distinguish between different cells in the same way.
Yeah. So this is a fairly large animal that I'm showing here, and each of these little dots is an individual cell body. And then if I zoomed in here, you might see some little lines. So those are your dendrites. So you can really pick out cell body and dendrites. So you can really image every neuron and what it's doing. Yeah.
Thanks.
Yeah, sure. Great. So anything else? Okay, awesome. So from here, I'm going to talk a little bit about some of the experimental work we're working on toward this goal of imaging all of our neurons and testing these sort of physical models. So what do we need for something like this to work? So again, the goal is watch synchronization of the whole nerve net from an initially disconnected state, and we're going to do that by imaging every neuron in a redeveloping aggregate. So toward this goal, we need a few things. First, we need our transgenic strains. So we need something that we can actually, properly image. We need to keep aggregates alive throughout this imaging process. And we need a microscopy method that allows us to image every neuron in a redeveloping aggregate. So from here, we'll just sort of focus in on these methods. So first, making our transgenic strain. I'll breeze through it quickly. The animal that we wanted expresses GCAMP, which is, again, a fluorescent calcium indicator, which shows us neural activity or when individual neurons are firing. We have TD tomato NLS, which is a fluorescent nuclear label that is fluorescent all the time so that we can track where the neurons are and what they're doing. And last is this NTR, or nitroreductase, which is an enzyme that catalyzes a prodrug, metronidazole, into a poison. The idea here being that we can do experiments like I had showed you before, where we knock out the neuron cell line. So to breeze over all the details, we express these 3 proteins under a single promoter, and sure enough, have our animal. This is like 8-9 months of work condensed down to a half a slide. Lots of injections and screening and suffering. But here we have it, right? This is our transgenic animal that we'd like to use. So starting from here, we'd like to be able to keep these aggregates alive throughout this imaging process. What we had found was that sort of the state of the art, all the stuff you've seen on aggregates so far, people are taking this aggregate, squashing it in this chamber that's between 2 pieces of tape that's about 200 microns apart. And if we take an adult animal and do this, what we find is that about 14 hours, it's still healthy and moving around, and you can see the neurons are still firing. But by 16 hours, believe it or not, this is a movie, but nothing's happening. Everything is dead. So the adult animal only lasts about 16 hours in this sort of chamber. and the aggregates last even less. The reason being that aggregates, their cells are already under quite a bit of stress, and so squashing them in these chambers is certainly not good for them. So we're not going to be able to image the entire reassembly process over several days. So one important observation was that Hydra and aggregates can survive for up to a month without any care in an open dish. And so our guess was that it was really oxygen that was limiting survival. So I worked with a really great, at the time, undergrad, Charlotte Mastrato, who's now at BU, making these perfusion microfluidic devices. I say microfluidic in quotes because they're really quite big, right? You're trying to trap an aggregate about a millimeter large. And what we had found was that we were using laser etching as a means of really rapid device development. I wanted to sort of emphasize here that we're using this very nice laser etcher to go from a drawing to a fully assembled, ready to use microfluidic device in like 20 minutes. So if you're interested in these sort of rapid device development, happy to talk about it. And what we had found with these chambers when we finally settled on a design is that you can trap a Hydra like this. You can still image all the neurons, and here you're imaging contraction. So you're seeing this burst of GCamp in the foot here. And in these sort of chambers, in contrast to these squash chambers, they can survive for a week or more. And they're quite healthy without feeding or anything. They just flow fresh media over them, solving our sort of oxygen availability problem. But we still had one issue, was that in these chambers, aggregates would also often get dislodged or damaged. And what we found by looking a little more closely is that when we look at the flow, there's lots of backflow, there's lots of stopping. And for these very soft aggregates, this backflow would just rip everything apart. And so, and then we had bubbles that would come through that would completely wreak havoc and destroy everything. So we drew inspiration from these sort of IV drips where fluid drips in the top and then collects in this chamber and then flows out of the bottom. And this flow, we had guessed would be quite a bit more smooth. We built a system that looks something like this, where our perfusion pump, instead of going straight across our aggregate, now goes into this sort of IV drip-like chamber, which then flows across the aggregate and then recycles back through this reservoir. What we found is that in this system, the hydrostatic pressure at equilibrium gets balanced by the resistance through the chamber, and you can go through and solve some Bernoulli's law to tell you what the diameter of your tubing should be and what the thickness of your chamber can be for a certain height of your fluid. We found it works quite well, and at some point that the flow in from the perfusion pump equals the flow out, but that we have the added benefit that bubbles don't go through, and that the flow through this system is really steady because you're averaging over all these little drops as they go into this chamber. And so this looks something like this afterwards, where we have this very nice, steady, even flow. And so what we get out of this is that the Hydra are subject to a much more stable flow, and we can take our aggregate immediately after forming it, put it in this chamber, and what you see is that some of the cells are lost, lots of dead or dying cells, but we're able to capture this thing really early and start to see the formation of these sort of coordinated motion of this aggregate. And then we went ahead and took this aggregate and loaded it onto a fluorescent microscope, where we see that by 22 hours you can start to see these neurons and some coordinated motion, and image it all the way through at 1 second intervals all the way through to 75 hours, and really see now that there's many more neurons. We see the formation of, right here, tentacles, another tentacle, a mouth. So this thing is now started to fully form its body plan and nervous system. So we have this really nice system where we're able to capture and keep healthy and image our aggregate right from the very beginning. And so the last challenge that we had set for ourselves was to be able to image every neuron in the redeveloping aggregate. And so what we needed was a system where we can image a sample that's size, about 1 millimeter by 1 millimeter by 200 microns. We need to be able to capture it at dynamics of about half a second so that we don't miss any calcium signals. And we need to be able to image this for multiple days and up to a week at a resolution of about 10 microns so that we can differentiate one cell body from another. So this is a lot to ask of a microscopy system. And so what we're working with is a setup called Fourier light field microscopy, which looks something like this. So on this side, you have a pretty standard fluorescence microscope, where if you are used to looking at microscopes, you have can recognize all these parts. You have your objective, mirror, tube lens, and then normally you'd have your camera right here. But in this setup, you have a microlens array, which effectively is just one extra lens, and this little lens array, which is exactly what it sounds like. It's just a set of lenses. In our setup, it is a 3x3 array of small lenses. And so it's a fairly simple change to a standard microscope, but what you get out is really fantastic, right? So this is one of the earliest works on this Fourier light field microscopy, and what you see is a zebrafish embryo brain being imaged at high space and time resolution, again, just with this very simple change to a traditional microscope. So how does this thing work? We have our objective, our tube lens, our Fourier lens, our microlens array, and then here at the end we have our CCD, where this is from the side, and this is what you'd actually see from your CCD. If we have a point somewhere, this will then propagate through this system and show up as distinct points on your CCD. In our case, because we have 9 lenses, we'll see 9 different points. And if you move this point around, you get what you'd expect. The point would move, up, down, left, right on this CCD, just like you'd expect. However, where the real magic comes is when you have a point that is behind this initial point. Now, in a standard fluorescence microscope, you wouldn't see this blue point because it'd be covered by this red point. But what happens in a Fourier light field microscope is that it propagates through in a slightly different way, and you'll see on the off-axis lens elements, you'll find this point. So what's effectively happening here is that the position in these 9 images is encoding your depth information, or equivalently, your point spread function is telling you how far away you are. And this is sort of the same principle as stereo vision, right? You have 2 eyes that are picking up 2 slightly different images, and your brain is piecing together the depth information, how far away things are. And why this is really great is because it's scan-free, right? You can image as fast as the camera can take pictures. You don't have any moving parts, and you're getting all this depth information. So if we take our Hydra, put it in this Fourier light field microscope, we can go ahead and reconstruct and get our full nervous system, where we can see now every neuron in this animal. And so the challenge really is in this reconstruction step. So for some object in X, Y, and Z, when we take an image of it, we're effectively convolving it with this point spread function. And then when we look at it on the CCD, we're projecting it down into this 2D image. And so this would work something like this, where you have your object that you're convolving with your point spread function and summing over to project onto your CCD. What we can do is we can measure this image, of course, just by taking the picture. We can measure this point spread function using some pretty standard methods. And what we'd like is to back out this object. And this is a pretty common procedure. It's just deconvolution, except it's slightly different than standard deconvolution, which is that in normal deconvolution, you're going from a 2D image to a 2D image, or a 3D image to a 3D image, and here you're going from a 2D object and a 3D object and trying to recover a 3D object. So you're using sort of a Richardson-Lucy based deconvolution, which is a very standard method, but you can't use any of the packages that already exist because it's a little bit different than everything else that's out there. So any questions about any of this so far?
Ray, I have one question.
Yeah.
So can you let me know the actual resolution of this? That is the first question. And the
Sure.
second question is, it seems you use 3x3 microarray lens.
Yeah.
And is that there is some particular reason for that? Or if we change it to more microlens array, is that can be the actual resolution or lateral resolution can be improved?
Yeah, so both great questions. So first, in the setup that I'm using, because I want a fairly large field of view, I have to sacrifice resolution. And so the XY resolution is something like 5 microns, and the Z resolution is always worse. So in theory, it could be 8 microns. In practice, it's probably closer to like 12 microns. But this, of course, changes as you change resolution and different features of the microlens array. And that gets to your second point. So why not use more lenses? More lenses will help you for sure. You will get better axial resolution. And in this original paper back here, where they were looking at this full zebrafish brain, they were using, I think, 27 lenses put in a very specific arrangement. So why use 3? Well, for us, the resolution that we're getting in the field of view we're getting is good enough with 3. So we're sticking with that. It would be nicer to have more lenses because you would get better resolution or better depth of field. And the other side of it is that there's really not a good source for buying these sort of lenses. So the one that we have to cut glass and then mount ourselves. So the more lenses you do, the more challenging that process is. Yeah. Does that answer your question?
Yeah, thank you.
Sure. Okay. So then on to this reconstruction step. We went ahead and looked around at what sort of algorithms are out there. And what we found is generally 3 algorithms that have varying speeds. So the first and the most simple is one that was called Olaf, which, on my setup, could reconstruct an image in about 2,000 seconds, but was written really nice, open MATLAB code, but wasn't at all optimized for speed. There's a company out there that actually makes these sort of Fourier light field microscopes, and their system for reconstructing they call DoIT, and we found that it was running about 200 seconds per image, so it's still fairly slow, especially if you're imaging, a Hydra for a week, right, at half a second resolution. I mean, this is going to take years to finish your reconstruction. It runs in C, and the code is proprietary and confidential. And so if you want to, edit things, it's very, they won't let you do it. On the other hand, there exist methods like convolutional neural networks, which are quoted to be, at under a second. They're sort of a black box like all these machine learning methods, which is that when they work, great. When they don't work, it's really hard to debug what's going wrong. And you need to train a new algorithm for each data type. So if you change magnification, or if you change, from an aggregate to a polyp or a different animal, there's no guarantee that you'll actually still get out something that looks all right. So our idea initially was fairly simple, which is just take these sort of algorithms and try to optimize them as well as we can and see how good we can get. And that's a fairly simple idea, so why hasn't it been done before? And I think it's just because there's sort of a mismatch between people who develop these sort of tools and the experimentalists who might want to use them. So, I would think that the appeal to most experimentalists would be speed, right? You can take an image and right away see a 3D volume. But if you look through the literature, most of the development focuses on boosting resolution, right? Trying to shave off half a micron in X, Y, or in Z, or to try to give you a greater field of view. So I think there's just sort of this mismatch, and there's not really a lot of folks working on optimization. And in that sense, it's sort of low-hanging fruit. So the details don't matter. I started with some code. I did some basic optimization and got it down to about 3 seconds per volume. And then where the real magic happened was bringing on folks at Johns Hopkins Scientific Software Engineering Center. So real software engineers who were able to take the code that I sent them and accelerate it down to 140 milliseconds. And so for our use case, this is effectively, real-time reconstruction where we can take a picture and see a 3D volume right away. We are publishing this as an open source software package. We're releasing it in Python, in ImageJ, so you can put this right up to Micromanager if you have Micromanager and see these volumes, and also as a Dash application. We're calling it open source Fourier light field reconstruction, or OpenFLR, and it will live here in probably the coming week. So if you're interested in these sort of light field microscopes or these sort of methods, it's all open source, so feel free to play around with this stuff or talk to me. So why do I spend so much time talking about Fourier light field microscopy? I think that it's a really interesting method, and now that we can reconstruct volumes pretty quickly, I think it could be quite useful. So some of the benefits of this method is that there are papers that have really fast imaging speeds, using some tricks with camera, but not necessarily tricks with the change in the setup. If you have this microlens array, you can get up to 1,000 frames per second. You can image volumes if you go to higher magnification of 100 micron, about 100 micron by 100 micron, at resolutions of down to 1 micron. You can alternatively have fairly large fields of view if you use lower magnification, up to a millimeter in diameter and about 400 microns thick. And another great benefit is you have small data storage size, because all the information in the volume is stored as a single image, right? So you can take 100,000 images and have them on a single hard drive, rather than if you have volumes of stacks of hard drives to save all this data. Of course, there's limitations. The sample has to be sparse, right? If things are really dense and objects are occluded by other objects in multiple lens arrays, it's going to be hard to reconstruct them. And there's sort of an exaggerated trade-off between field of view and resolution. I mean, if you go to higher magnification, you always lose some field of view, but it's particularly bad for Fourier light field microscopy. But it's been used on lots of really interesting samples that are, I think, broadly interesting to folks working in, soft and living matter, which is, bead tracking, dynamics in different tissues, and rapid tissue reconstruction. So there are lots of great applications. If you're interested in these sort of methods, I'm happy to talk. So where does this leave us? Well, we have now the transgenic strains we had wanted. We can keep aggregates healthy and immobile for these days or week-long imaging. We have this fast 3D microscopy to capture every neuron in the animal. And so I think we're really well established to start taking the data we'd initially wanted to. Right now, we're just working to integrate all these systems, and early on, we have this raw data of a Hydra moving around and tracking its neurons over time. So stay tuned. Hopefully, we have some really interesting data rather quickly. As an aside, since we have this sort of trainee hour afterward, I wanted to mention another project just in one slide that I've been working on, which is trying to get at the question of what are the molecules that govern nervous system self-assembly. And so what we've looked at so far is this top-down approach where we image every neuron and say, how does the nervous system structure emerge from imaging hundreds of neurons? And this is trying to get at it from another direction, which is if you have an individual neuron, what sets who it's going to synchronize with, or how does this neuron know that it's a contraction burst neuron and should fire with other contraction burst neurons? And so we've been using some excellent data that's been published, which is single-cell RNA sequencing, which tells us the protein expression in every kind of neuron across the animal. So we have 12 different neural types, and we can look and see what proteins are expressed in each of these types, and also correlate that with the neuron types that are associated with known behaviors. And so what we can do here is really, Hydra is giving us this unique opportunity to correlate protein expression with the formation of these synchronized ensembles and the behavior of the animal. And what we're finding is that it seems like gap junctions expression is correlated with behavior, meaning that contraction neurons, all the different kinds of neurons that are associated with contraction, express lots of different kinds of proteins from each other, but they all express the same kind of gap junction. And it's the same thing with rhythmic potential 1 and rhythmic potential 2 and all these other synchronous groups. And so what we're doing is using different methods to sort of block these gap junctions or do drug treatments to try to pick apart whether these gap junctions really are the driving force of self-assembly, whether they're encoding how a neuron chooses to make connections with other neurons, and how it chooses to not make connections with certain neurons. And so with that, I wanted to say that I hope I convinced you that Cnidarians are an excellent model for studying neuroscience and the physics of living systems, that we've solved our sort of major technical challenges by making these transgenic strains, developing this fast imaging platform, and microfluidic devices, and that we're really at a point where we're ready to integrate all these devices and test these sort of synchronization models. So if you're interested in Cnidarians, light field microscopy, synchronization, or really anything else, I'm happy to talk. I wanted to thank the folks in the Howard Lab, and in particular, Charlotte and Tarek, who are undergrads who worked with me on this project. Thanks to all of our external collaborators, and thank you for your time, and happy to take any questions.
That's great. Thank you, Ray. Do we have any questions from the crowd? Feel free to just
I have a question.
Yeah.
I'm just curious about when you mentioned the self-organization of the neurons, is it more the neuron cells are moving is the major cause, or it's just the signaling level that are clustered and reorganized, or a combination of these two?
Yeah, that's great. So we can measure both because we have this TD tomato and GCamp both, so we can track our neuron motion using this this persistent TD tomato signal, right? The red fluorescent, and then we can watch the activity using GFP, so this green fluorescence. So whether it's migration, whether neurons move to the place that they're supposed to be and form these groups, or whether they sit still and then branch out and make these connections. It's too early to tell, but that's for sure one of the things that we're really interested in. Or alternatively, whether they just differentiate in place, right? That's another option that is certainly possible. Yeah.
Okay, thank you.
Yep.
We have Daniel, I think. Yes, Daniel.
Yeah, two questions. First, a quick one, just being at Johns Hopkins, I was curious how this collaboration with the software engineers at Hopkins came about. And then second, since I know you've also worked with light sheet microscopy, I was wondering if you could compare the two methods.
Yeah. Great. So with Johns Hopkins, they have a partnership through, so the fellowship that I'm in, the Schmidt Science Fellows, they have a secondary collaboration called Scientific Software Center, where they work with places like Johns Hopkins. They have another center at University of Washington and one more somewhere else, where if you're part of this sort of group, you can apply to work with software engineers. Now, lots of universities have like internal things where you can try to work with these software engineers. If you're interested in that sort of thing, definitely give it a shot, because it is fantastic what these folks can do. I mean, you think you're good at coding until you talk to a real software engineer, and then it's so that's how that started. What was your second question?
Oh, to compare with light sheet microscopy.
Oh, yeah. So light sheet microscopy, I mean, it depends, right? I think it certainly gives you better resolution, and it's a little easier to interpret up front because you don't have this extra reconstruction step. I mean, depending on the system you're using, it can be slower. There are, tricks where folks are doing, very fast light sheet imaging. But there you're going to, fairly expensive setups and homebrew setups, and also now you're storing these very large data volumes. And so if you're trying to image, at half a second for, 7 days, I mean, you just got massive amounts of data to go through. And for us, especially for samples that are really sparse, You just don't seem to need it. Now, there are cases if you have very dense samples, you're stuck with light sheet microscopy. So, yeah.
Great. Thanks.
Yeah.
Suraj next.
Yeah, no, this is fantastic. I wanted to get some thoughts. Hear some of your thoughts on the development of the nervous system that you are currently working on, and maybe have some insight on. And I was wondering, as they start synchronizing, what you mentioned a lot of descriptions of synchronization, largely models, but also potentially mechanisms that were only purely neural. What are your thoughts on mechanical interactions, presumably at early stages, and especially given that you have these different subsystems that specialize over time? Are there ways of probing and learning behavior? And can you train and entrain subsystems and see how that emerges over time in the growing neural net?
Yeah. Great. So as far as structural goes, I think it's a very interesting question. One thing that's observed is that between like these two states, right, where you have stuff that's initially uncoordinated and desynchronized, to when you start to see synchronization occur, if you look at the individual neurons, you start to see outgrowth of the dendrites. And so as these dendrites grow, of course, they're starting to make connections with each other and explore the space around them. So there's certainly some interesting questions about how does this structure influence the synchronization, and how does an individual neuron, does it sort of explore the space around it through branching, or is there some guiding something that is telling it who it ought to connect with? Those are great questions that we just haven't looked at, but certainly would like to. And then your second question was on sort of learning.
Yeah. Particularly with the sort of specialization with different subsystems. And
Yeah.
initially when you're all unsynchronized, presumably everyone starts out very similarly, and then different parts of the nervous net that start specializing, and how much of that depends on the cues, the sort of sensory input it keeps getting, and allowing for that specialization.
Yeah. I'm inclined to think almost none in Hydra, because they are just extremely simple, that it's really all just the neurons have some barcoding that tells them who they are early on and who they ought to connect with and what muscles they ought to connect with.
So it's entirely centrally, so it's some CPG which is fully encoded is the view that you.
That's what I think. And the reason I think that is, well, so first, that you can take these things and develop them in the dark, and they still form substructures of the nervous system that are important for detecting light, right? So like these kind of things, like sensory inputs, seem to not really matter whether you culture them in the dark or in the light, or under flow, or in some confinement, or whatever, right? So you end up with the same sort of structure. The other side of this is that
sure, but can you maybe then refine that question and or that point, and then ask how robust it is, or its sensitivity? Is it still the same?
Yeah. No, I mean, those are good questions. Yeah. And I don't know. I don't know whether it's the same, or whether, you can do things like, really overstimulate these neurons and then, see them return to baseline, and these kind of questions. Yeah, for sure. There's interesting stuff to do here. Yeah.
Okay. Oh, thank you. Fascinating.
And nobody's really looking at these things. So, certainly.
Even more impetus to study them.
Yeah, for sure.
I see Anna has a question.
Yeah, hi. So I was just curious about the gap junctions. So you're saying that they express different gap junctions. So does this mean that when they come together, if it's a wrong kind, it can't form a channel, and if it's the right kind, it forms a channel, right? Is that how it works?
That's what we think. Now, we're still working to show that, and we're using different different techniques like antibody blocking and drugs to try to, block formation of certain gap junctions or expression of gap junctions in neurons where they normally wouldn't be. I mean, typically what you find is that we have these different in invertebrates. You don't have the standard gap junction that we have is called connexin, and in invertebrates they call it innexin. And looking at Hydra, if you look across these different neurons, they have like the body contractions. All the different kinds of neurons that are participate in body contractions all express one kind of gap junction. And the other thing that's fun about these sort of gap junctions is that they're known in many contexts to produce adhesive forces between compatible gap junctions. And really, if you can make an electrical signal and you can produce an adhesive force, maybe that's all you need, right? You just have these two interactions and your neurons sort of click together with the ones that they're supposed to connect with. Again, that's a conjecture, but that's what we're working towards.
Mm-hmm. And another question is that so neurons can move an electric field. So have you done anything with that?
Mm-hmm. Great. So I haven't, but there is, in fact, it was, let's see, this work here that was looking at, morphological transitions in this Hydra. They weren't looking at the neurons in particular, but what they were finding is that by what they were thinking was causing this transition from a sphere into breaking symmetry and becoming oblong was different electrical impulses across the body of the animal. And what they found is that by putting this thing in a potential in voltage, right, in a voltage cell, you were able to go from an animal that had fully formed a body axis and had tentacles on one side and a foot on the other. It would actually shrink back up and become a rounded ball again. And then if you turn off your electric field, it goes back into an animal. So I don't know what are the implications of the neurons in that case, but certainly there's huge implications in the morphology of the animal. Yeah.
Cool. Thanks.
Yep.
Do we have more questions? So I have a question. You're saying that they do not respond to external stimuli while developing, it's like predetermined, but I'm still puzzled by the size, the fact that they scale with the size of the system, or that the group scale with the size of the system. So could you just play with the initial aggregate that you have to kind of see the number of, maybe there is a critical number of mass or neuron that can develop into something, and then do you have this bifurcation in the system?
Yeah, for sure. So what you get is that if you have an aggregate that is about, I mean, the magic number seems to be about 100,000 cells total, you'll get about one animal out. Now, as you start to add more cells, so if you do 200,000, what you'll get is something that looks kind of like this one, which is that you have like a head over here and a head over here and maybe some other tentacles, and this may be a separate body. And so the number of cells you add, really there's some way that it sorts itself into clumps of about the right number of cells and then breaks apart. There's a little variability from one to the other, but yeah, effectively, that's what you get.
Okay, cool.
Yeah.
Do we have any other questions?
Hi, can I ask a question?
Please,
Yeah.
yes.
Yeah. Hi, Ray. Nice talk. So I see you mentioned there is a lot of model about the synchronization. I was just curious, usually the synchronization models, will they include something, the feedback?
Feedback, so you're meaning like from one neuron to another, that sort of thing?
Yeah,
Or
something like that. Like when the neuron works together, are they going to like change from this pattern to another pattern, or they just keep the pattern doesn't change and continue moving?
I don't know. So if it's the case that everything's predetermined, then and neurons just connect up with other like neurons that are expressing the right proteins, then it's a little more boring than these sort of synchronization models, right? Because if you play with these models and you have lots of identical oscillators or integrate then fire or whatever, you can get some pretty non-trivial states. But if you have just groups of, individual different oscillators that only interact with like kind, well, then you kind of just have this boring, everything synchronizes together. There's still questions about like, if you have a group, one group that synchronizes in one area and one group that synchronizes in another, and then they start to meet each other and merge together. And that's been seen in like zebrafish embryos, right? Along the spinal cord, you'll have synchronous domains that emerge and then all synchronize together. So it depends how the data comes out, but certainly, I mean, there's stuff to think about there. Yeah.
Yes, so I was just wondering whether those model will include this kind of possible mechanism, like they have some parameter to describe this behavior.
Yeah, so some do for sure.
Mhm.
Some have, polarity built in, and some have, different connectivity built in, and the most simple ones don't have any of that, right? And so the question is, how simple can you go and still describe the data? And I don't know.
Okay, thank you.
Yep.
Can I ask another question as well?
Yeah.
Yes.
Yeah, I think again coming back to this point of the synchronization in terms of like, and then guessing if there is a role of the mechanical feedbacks into it. So my guess is like the neurons would control how the muscle fibers in these contract, and therefore there will be mechanical synchronization as well, along with the synchronization in terms of the signals in the neurons as well. So
Yeah.
is there a role of like then mechanical synchronization as well, which feeds back into the like, and if there is like good enough reason to believe that from looking at the experiments?
Yeah, I mean, that's a great question. So I really don't know.
No, yeah.
You would think that there must be something. And we could certainly get at that because like there's these sort of lines that already
Yeah,
exist.
exactly. Like the
Yeah.
system is really, really fascinating.
Yeah. So there's like these lines
Observations.
where if you have you could express like a red protein that becomes bright when the muscle fires in, let's say, the ectoderm, and then you could do a green protein in the neurons, and you could see this sort of interaction, right?
Yeah.
That's a great question. Now, what you see is that when the neurons fire, the muscles are extremely responsive. I mean, they're right away.
Right.
So my initial guess would be that you have these gap junction connections, that the neurons are connecting with the endoderm and ectoderm also through gap junctions. And we know for sure that all the muscles is connected together with gap junctions. So maybe it's just all one big electrical circuit.
Right. Like as in, then there would be directionality, like the muscles can provide a specific directionality in which the signal propagates. Is that
Exactly.
idea?
Because because if you have gap junctions, everything's electrically connected, right? It's bidirectional. So
Right.
if you have synapse, right, where you have a chemical synapse, activating a muscle, let's say, then it's from neuron to muscle. But I don't know if that's the case here. I'm not sure.
Thanks for the call.
Yeah.
Can I ask one minor question?
Yeah.
So I'm curious about the sparsity condition in the Fourier light sheet microscope.
Yeah, sure.
So I think if there are too many light sources in field of view, all the engineered image in the image sensor should be overlaid a lot, and in the limited dynamic range, that may cause the decrease of accuracy of reconstruction or decrease reconstruction performance.
Yeah.
Do you have any idea about that? So how much the reconstruction performance will decrease because of that not sparsity?
Yeah. So in the it's hard to get an exact number on it. In the setup that we have, what we've done is that we've set it up so that the different lens array, the images from each of the lenses just barely touch each other, right? So you won't have overlap between two images. But I agree with you that if things are very dense and that you have a lot of overlap between your individual images, it's going to make reconstruction quite a bit harder. And I mean, that's what they had done in this original paper where they had, like 27 lenses. To put 27 images on your CCD, lots of them were overlapping, and they had a lot of tricks to try to, clear this up so that there wasn't so much overlap from one to the other. But, yeah, that for sure, that's a problem. But quantifying how much is it a problem and how much it affects the resolution, I don't know. Yeah.
Okay, thank you. Yeah.
Yeah.
Okay, I will stop the recording at this point, but if we have more questions, you are welcome to ask.
Sure.